Hello, everyone. Thanks for joining today. Thanks for taking time out of your busy schedules to learn about pests and disease. Before we get started, I just wanted to go over a few housekeeping rules. So first, if you haven't already, please email me your license numbers for CEUs. So if you have an arborist, ISA license, please email me that. If you have a restricted use pesticide license, please email me the category number, the license number, and the expiration date. And if you have an LICT certification, just email me that you have one. I don't need the number. After that, please, once the presenter starts, please make sure to mute your speaker, your phone, or your microphone so that we can hear Brian Bushe, our presenter. And in the middle of the presentation, I will be doing a roll call. I'll ask you to please, in the chat, which if you look at the bottom of your screen, you click on the chat, and please enter your full name so we can give you credit if you're here for CEUs. And also, if you have questions during the presentation, please enter it in the chat and we will review it at the end, the last 30 minutes. All right, without further ado, I'd first like to thank the Maui Association of Landscape Professionals, who is the host of this webinar. And first and foremost, I'd also like to thank Mr. Brian Bushe. He is the agricultural diagnostician for the College of Tropical Agriculture and Human Resources at the Agricultural Diagnostic Service Center in Hilo. He's very experienced and has taught me a lot about pests and disease. And yeah, that's about it. Brian, are you ready?
Sure. I'll probably need some help getting this to actually run, but I am ready. Hi, folks. This is kind of new. I usually give these presentations in person, but I guess we're going to try and do a little bit of high-tech stuff here with someone who's Cro-Magnon on the computer. So I'm going to ask Hannah's help. As Hannah said, I work here in our Hilo office for the extension service. And it's actually a rainy morning, which isn't all that unusual for us here in Hilo. I don't know how it is for the rest of the state, but we did have nice weather yesterday. So I go share screen, right, Hannah?
Yep, at the bottom, share screen.
Okay, and find my presentation. I'm also working with a borrowed laptop from someone else, and it's rather old, so it's very slow from beginning. There. Can everyone see that? Can you see that? Okay.
Yep. Thank you.
Thank you. Okay, so as I said, I'm here in Hilo for Hawaii County, and that's the island that I represent. I work for the Agricultural Diagnostic Service Center, or the ADSC, and I am a satellite here on the Big Island. And I was hired specifically for growers here on Hawaii County, in Hawaii County. So I do work on insect and disease problems here. Let me give me a little bit of time to get myself situated here. I'm going to actually read from my notes. We thought this would work best for Hannah and myself. Everybody can hear me, I think, huh? Yeah, Hannah?
Yes.
I'm clear? Okay.
Yep.
Borrowing my son's gaming microphone because my wife tells me I mumble. So hopefully you can hear me throughout the meeting. Otherwise, Hannah, maybe you can give me some sort of warning. Okay, thanks. Sure. Okay, so the goal here, the reason the Farm Bureau pursued having a position here in Hilo is because of the amount of agriculture that occurs on the island. And so what the industry really wanted was help with their insect and disease problems because, as you all know, they can become critical problems pretty quickly. And so this position began in 1989 when I started, and the goal has always been to provide quick, accurate service, and I've worked very hard to do that. So can I, let's see, how do I progress? Just with enter, I guess. So services are also done at our main campus on Oahu. They provide not only the insect identification and plant disease work for the other islands, they also provide the feed and forage analyses. Okay. Chemical analyses of soil, plant tissue, irrigation water, nutrient solutions. Hmm. Let's see, I am doing something wrong here. Can you see all that on my screen? No? Can I close that?
Mm-hmm.
You can see the
Yes.
participants. So I should close this.
No, I don't see the participants.
Okay. Yeah, it's right over my screen, so I can't really see what I'm showing you.
You should be able to move that
Am I?
participant now.
Brian, if
Yes.
you at the top where you see all the faces, you
Yeah.
hit the minimize, the hide thumbnail video. The
Yeah, I'm
minus
hitting
sign.
it. Yep, I'm okay, there we go. There we go. Okay. That cleaned my screen. Why am I going backwards?
You can
Come
use
on.
your arrows at the bottom.
Yeah, I'm doing that.
Hit enter.
Hitting enter. Seem to have frozen my laptop. There we go. Okay, and the other thing that we do with the Diagnostic Service Center is we sell UH developed fruit and vegetable seeds. Okay, but it is a fee service. Um, we do actually uh, uh, charge for these diagnoses. And, we really are quite a bargain when you consider what the costs are for medical lab technologists or diagnostics, as well as for your pets, right? And if you were to actually look at the fees other colleges charge the general public and growers throughout the US, you'll see that Hawaii is really cheap. And we also charge request payment before the test initiation so that we don't have to be bill collectors. I run the lab here by myself, so I couldn't possibly do it. Okay, and then there are many CTAR publications regarding sample selection, preparation, submission available on our website. And the reason I bring this to your attention is because the sample you collect uh, will determine the accuracy of the results you receive. So you really want to make sure that you're collecting the proper, in the proper method and treating that sample with care so that it's not ruined by the time it gets to the respective lab. And we have a publication created. You see a link there at the bottom of the slide as to where you can actually find this information. And it froze again. There we go. Okay. So, Hannah, I can't really see anything. And if anyone needs to contact or if you want to stop me or anything, please tell me. Okay.
Will do, Brian.
Thanks. Okay. So, well, let's specifically talk about insect or arthropod. We're including mites here. So mites aren't insects, right? We all need to know that because they're treated differently. So we'll call them arthropod and disease samples for submission to the Agricultural Diagnostic Service Center. The thing to remember is, again, it's all in the details, okay? The best result you receive is based on how good the sample is you submit. Okay, so uh, again, the accuracy is only as good as the information you provide. You know, sometimes the agent is not able to be in the field to see what you got. You walk in the sample, and certainly myself or the diagnosticians at Manoa aren't in the field and aren't able to see what's going on. So the sample must be representative of the symptoms and the severity in the field. So they must contain the right material. They must be fresh and in good condition, and then getting it to me or getting it to the lab rapidly could be critical. So sending it on a Thursday is never really a good idea if you're mailing it from an island to another island, because they tend to get stuck somewhere and they rot. And then the accuracy of your source data and your information is also important. And how, where, when was the sample collected? So what do I mean by that? Well, you want to look at comparative things, healthy versus sick. Make sure that you're not dealing with a sport or a chimera or something that is genetically mutated that a lot of the ornamental industry will use, right, in planting different crops. So make sure you know what a healthy version of the plant looks like. Um, you want to be able to describe the symptoms, the development of the symptoms, and then the progression, right? How it occurred. So you observe patterns. Was it a rapid or a gradual occurrence? What was the distribution spread? What's the history of the problem? Uh, Seldom ever is it like it happened overnight. It was all overnight, because it just means that it probably wasn't observed, and it occurred over a few days to a week. And then the growing conditions. Oftentimes, especially with home gardeners will see folks trying to grow apples at the beach, right? So, is it just a plant that's growing out of place? Is it a sun-loving plant grown in the shade, or is it a shade-loving plant that's been put out in the sun? Or perhaps the canopy cover has been cut back, causing some other injury. And then, of course, what are the cultural practices? Is it fertilized? Has it been fertilized? Is it under irrigation? What sort of sprays have occurred? And the thing about the college here is that we're not regulatory, so you don't need to be hiding what you're spraying. We need to know what happened to help you figure out how to proceed and go forward. And so how do you remember all that? Well, luckily we have a form in the college, in the extension office, for the Diagnostic Service Center. It's called the Form 357. It's up in the upper right-hand corner of the form here. And it will help you recollect and provide information that we need here in the lab, or the agent needs to help assist you. So here's some examples of just what a good or a bad submission looks like. So if you look over in the, if I move my mouse, can you see that, Hannah? I think.
Yes,
Okay.
Brian, I can see that.
Okay, so here in this image is a cypress, and the problem that the grower faced is some dieback on the leaf tip. And so this was an excellent submission, because we could see the progression. So as it started to die back, the tissue we would isolate from is right at the juncture between the healthy and the dead tissue. And so that would actually tell you where that pathogen is, right? So it's actually dying back. If you were to just break off the dead tips and submit that, then I would probably recover saprobes, or organisms that are involved in cycling back into the earth. So that would be a wasted analysis, because I couldn't tell you what happened. All I could tell you is that something has come in and started to destroy it. So Oliver has entered the room. I can't see the image under that now, Hannah. But that's an example. The image over here on the right is an example of a poor sample that was left to dry out. Here we go. Left to dry out, and uh, it was, not so easily identified a cause. And so you don't want to do that. Placing it in a plastic bag without moisture, like don't want to put in a wet paper towel in a situation like a succulent. But you can do it with a plant like this cypress. So an example of that here is uh, this sample here. Below is um, a branch tip in which they put in a dry paper towel to absorb any moisture. So that's what you would do. Leaving it exposed like in a brown paper bag and mailing it, they tend to dry out and look like this, and it's difficult to work with. And then the opposite is true where they place a wet paper towel in with this is a flowering plant, so pretty succulent, a lot of moisture content, and it simply rots. And it's a little bit more difficult for the lab to tease out the causal organism because of all the saprophytes that will grow. Saprophytes are free living organisms that start to cycle or break down plant material into dirt or into compost. So yeah, not a good idea to put in a wet paper towel. So this is another really good example here on the bottom left of how to submit a plant sample. And sometimes that's the best idea. If you can submit an entire plant sample like we did here with this potted plant, it gives us the opportunity to look at not only the foliage, the stems, but also the growing system. What do the roots look like? What's the pH and salinity of the soil? That will often tell you many of the issues you have in the upper canopy. Sometimes the salts might be high, or the roots are rotted, or perhaps there are nematode problems that you need to address. So in this example, they've actually tied off the soil to keep it from getting the foliage dirty, and that really helps to reduce this type of rot that we see here. And then this is a nice example of how to package turf. Just wrap it up in a newspaper into a Ziploc bag. This here is really an ideal situation where this is a local grower, a chrysanthemum grower, and the problem he was experiencing, as you see, is dieback. And he actually gave me a range of symptoms. So I got to see, where's my mouse here? I got to see this healthy plant and then progressions as it died back. And this allowed me to look at not only the flowers and foliage for issues, but also the roots. And in this case, the plants were infested with Western flower thrips. And because of that, we tested it for tomato spotted wilt, and that was the problem. This actually came from Maui. This is a sample that was sent in a wet, soiled plastic bag, and this is what I received. And the complaint was that the plants were rotting, and I could agree. I concurred. That's what they were doing. So I couldn't really recover any problem other than they were rotted. We also have a publication that helps to work through how to submit samples to the labs for diagnostics here at this publication. Enter. Sometimes the issues aren't disease related. Okay, we can actually create our own problems with different methods for causing disease or causing an abiotic disease, which is simply mistaken planting. So an example would be this plant here was planted too deep. You see here is the crown where it should have been planted, but it had been buried. And so you've got all this adventitious shoots as the plant is trying to recover, and it eventually succumbed to root rot. And in the center here we see another real common problem where the plant was pot bound and put into the ground, and it grew for a little while. Oops, we've got automatic lights here. Where it grew for a little while, and then eventually it burned itself out and died. So it's always a good idea to prune these roots before you plant them, so that they won't continue growing in a circle. And then, of course, other cultural things like the irrigation was turned off, or we've got flooding. And we've also got a publication that will talk about some of these problems and how to recognize them. So here's another good example of what happens when we simply um, uh, over are overzealous in the way we grow a crop. So this actually is a lychee planting out on our Hamakua coast, and they put in 5 acres of lychee, and they were doing really well. These are all air layers, so this here is the recommended planting depth, and she planted it a little deeper than she should have, but they were doing fine. The plants were taking off, and she thought, you know what, I love my plants, and I am going to go ahead and improve their chances of survival by mulching. And so she placed mulch right up against the trunk here, right up to the first cross branch. And unfortunately, it killed the plants. And so this is a good example of the title to that song by Queen in the late 80s, that too much love will kill you. So yeah, we can love our plants so much that we cause these types of problems. And unfortunately, it's pretty common. She saved about 50% of her crop by pulling back the mulch, but of course it was a lot of excess work. Okay, so having said all that, hopefully I've given you guys a good idea of what to look for and how to go ahead and collect your sample and submit it. And you know, you are the eyes and the ears in the field. You know your crop best. You know everything a lot better than any of us at the college do, because you're so familiar with your crop. So sometimes just taking all of this stuff into account will help you uh, isolate, or at least uh, um, come to an understanding of perhaps what might be causing the problem. And then, you know, you can incorporate the use of our labs or our agents to help you confirm or figure out what might be going on, talking it through. So unfortunately, the biologics are a big problem in Hawaii. We all know we don't have a winter here, you know, so there's no freeze, there's no starting from ground zero. Their pests are here to stay and are happy, and are a problem for us year round. So what I thought we'd do to begin with is perhaps look at the most common problems that I come across here in the lab. Some of the more common insects and diseases that I think you will face as landscapers. And I wanted to start with the group, begin with an order of insects that are commonly observed in a landscape and that cause many of our problems: the mealybugs, the aphids, the scales, and the whiteflies. So first off, the mealybugs. We see here in this upper left corner citrus mealybugs. They all kind of look alike, right? They're all little fuzzy white balls of cellular material, but they can cause a lot of problems, and it's not so as important in the landscaping industry as it is in the nursery industry, because in that they are not quarantine pests for you all, but they are definite quarantine pests for the cut flower producers or potted plant producers, because they can't send plants with insects, right? So citrus mealybugs are a problem on things like gingers and palms and citrus, of course. The palm mealybug primary host is our palms. This Roseochus root mealybug is a problem on oleander, hibiscus, and palms. Tends to be when the plants are pot bound down in the pot. Again, this becomes a real big issue for the nursery industry because they're not obvious when they ship them. You can't really see the roots unless you pull them out of the pots. The coconut mealybug, very common on palms, fig, bird of paradise, coconut, its namesake. The pink hibiscus mealybug likes hibiscus, but it sure likes a whole bunch of different crops, including things like papaya. So they have a wide host range. And this Rhodes grass mealybug is a problem on poaceae or the grasses. Aphids are also quite common in our landscape. A banana aphid will feed on floral gingers, xanthosoma, cardamom, heliconia, tomatoes, taro, calla, costus, a whole bunch of different crops. The thing that we need to remember with many of these insects in this order, such as the mealybugs and the aphids and the whiteflies, are that they are viral vectors. So a viral vector is an arthropod because mites also spread viruses that spread the virus from plant to plant. So we're going to talk a little bit about that in a little more detail in the next slide. But I did want to show you that there are a whole bunch of different aphids in Hawaii. I mean, the book of aphids in Hawaii is about that thick, so there's quite a number of them. I'm just sharing with you the more common ones here. The cotton melon aphid feeds on a lot of different plants, including protea, tea. It transmits the papaya ring spot virus and the watermelon and cucumber mosaic disease. The fringed palm aphid doesn't look like an aphid at all, right? It looks more like a scale, but it's actually an aphid. It feeds on different palms, heliconia, gingers, calla, various orchids, and it transmits the banana bunchy top virus. The oleander aphid or the milkweed aphid feeds on oleander. Desert rose, vinca, stephanotis. It primarily transmits the papaya ring spot virus. And the orchid aphid, the preferred hosts are orchids here in the state. So as far as this vectoring of viruses, let me get my notes. So as we've all learned in this age of COVID-19, a virus is not a living organism. It cannot exist for long outside a susceptible host. Once inside the host, the coronavirus requires help to spread between hosts. And we now know that spread of this respiratory virus is through our uncovered coughs and sneezes. So plants don't cough, right? Or sneeze, so viral spread requires outside help, such as from an insect or mite, which we then commonly call a vector. So they're vectoring the virus. Vectors are able to transmit viruses in a few different ways. One way is non-persistently, in which the virus is retained by the vector for a short period of time, usually a few hours or less. An example of that would be papaya ring spot virus. Another method of transmission we call persistent describes how the virus is retained by the vector for long periods of time, often weeks or months. Some are vectors for life, such as tomato spotted wilt, which is vectored by thrips. Plant viruses can also be transmitted semi-persistently, which falls between the two, non-persistent and persistent. And an example of that would be banana bunchy top virus. So aphids are the most common vector of plant viruses. In order to better explain how viruses are transmitted, we thought we'd share this short video clip that better explains the process of how the aphid vector transmits the beet mild yellowing virus to sugar beets in a persistent manner. Sorry, the video content isn't that great. Let's see if this will work, Hannah. And we're gonna do it one more time. There you go.
If, on the basis of feeding probes, the aphid has recognized the plant as a suitable host, it typically moves to the underside of the leaf in order to start prolonged feeding. During feeding, the aphid stylet bundle reaches the sieve tubes, in which the beet mild yellowing virus is transported in the infected plants. The virus particles are passed through the aphid's food canal, along with the sap taken up while sucking upside down, into the esophagus, the stomach, and the gut. The virus particles cross the lining of the midgut and in this way enter the aphid's body fluid, its hemolymph. The virus particles circulate in the body cavity without multiplying. On their way, they also reach the principal salivary glands and the neighboring accessory salivary glands through which they enter. These viruliferous aphids transmit the beet mild yellowing virus from plant to plant along with salivary secretions. The aphid often remains a virus vector for life. For this reason, this type of virus transmission is termed persistent.
Okay, good. I hope that was somewhat educational. It gives you an idea of how important it is to control these vectors. So maybe not on the crop, but perhaps on the neighboring weeds, because they are able to spread these viruses. Did that work, Hannah?
Yes, great job, Brian.
Okay, it was kind of a blurry video, but I'm glad my son helped me figure out how to do that yesterday. So kudos and props to him. Okay, so take a look at scale insects now. Most scales are polyphagous, which means that they feed on a bunch of different plants. Okay, so they're seldom very specific, but there are species that are. Here in this upper corner, we've got the cockerel scale. This host plant include bird of paradise, oleander, azalea, plumeria, coconut, mango, kukui, cycads, and palm, just a lot of different things. It's known to infest over 100 species of plants. This phantasma scale is relatively new to the state. It really likes Araceae, which are the coconut, manila, aricas, the different bottle palms and such. Kamani, shower tree, heliconia. Boy, long list, right? Lots of different things as well. Easy to identify, with these stripes on them. Coconut scale really likes coconut and other palms, but we'll find it on banana, bird of paradise, breadfruit, mock orange, plumeria, lots of different things as well. Tea scale, also very nondescript, brownish scale, can't really identify it visually except by the host sometimes. It does feed on tea. It also likes anthurium, banana, coconut palm, dendrobiums, hala, hibiscus, lots of things. The cycad scale is one of those that is rather specific. It will feed on cycad and related families. The green scale also got a very wide host range. So if you notice here, I was talking about the armored scales. So these scales tend to be more like an opihi, where you can actually peel off this covering here, this white covering, and all the eggs or the juvenile stage of the scale is present. The juveniles are called crawlers. Here's an example of what I'm talking about. And the crawlers actually have legs, and they will boogie around, walk around, and look for another place to set up shop. They never move far from home, though. They kind of settle down right next to mommy. So this is mom, this is dad of the cockerel scale. They look different. So it's sexual dimorphism. They look different. This here is the female. She's rather large. And if you were to peel off the covering and you manipulated her body, it would be difficult to remove her. And that's because her stylet, or her mouth part, is fibrous and it extends down into the leaf. Sometimes you'll actually see where that stylet is in the tissue if you look really close, because it turns all chlorotic or necrotic right around where they're feeding. So if you were to manipulate this and remove it, you'd find that you'd have to actually forcibly pull her out or break off her stylets to get her out of there. So they're locked in there. Once they settle down to feed, they never really move again. Whereas the soft scale, I was talking about the green scale, this one has no shell covering. It will actually lift right off when you actually manipulate the scale. So you'll notice that it's all one piece. And like the armored scale, she does have a stylet that is placed down into the tissue to feed. And these soft scales, more so than the armored scales, are feeding at such quantity. They are trying to obtain protein from the plant. And in order to do that, they've got to process a lot of carbohydrates. So plants photosynthesize, right? So they're creating these carbohydrates or sugars within their system in order to grow. And as the scale is feeding on the plants, they are sucking in a lot of those carbohydrates, which they have to void. Okay, and when they do that, they void a sugary substance called honeydew. Okay, and that actually will cause a lot of observable damage, or not damage, observable coating of the leaves with a mold growth called sooty mold. So it's a fungal grouping of fungi that will grow on this sugary substance. So getting back to the green scale, this scale feeds on a lot of different ornamental crops, include anthuriums, gingers, orchids, gardenias, plumarias. It just likes a whole lot of different things, as does the hemispherical. Feed on ferns, mamaki, protea, orchids, mango, a whole bunch of things. The Niger scale as well, bamboo, coffee, cotton, lots of different ornamentals, even pineapple. And then the barnacle scales, the wax scales, such as the barnacle scale or the red wax scale or the stellate scale, the fig scale. There's 4 of them in Hawaii now, and very easy to identify by their appearance. So we talked about that honeydew, and I'll get right back to it. But before we do, let's talk about whiteflies. The 2 real common ones and ornamentals that you'll probably see in the landscape are the spiraling whitefly and the giant whitefly. This Bordard's nesting whitefly is brand new. We just found it last year here on the island, and it's unusual in that this is the native hibiscus, Waimea. It's unusual in that it's on the upper surface of the leaves. If you notice, the typical whitefly feeds on the lower leaf surface. And the reason for that is it protects it from the rainfall, right? So they don't get washed off. But this Bodar's nesting whitefly feeds on the upper surface. So pretty cool and unusual if you're an entomologist. But yeah, it's a problem as well. So the spiraling whitefly has been here for a while, since 1983. It got to our island in 1992, and it's got a pretty wide host range. You see it here on Plumeria. The giant whitefly came into the state later in the 2000s, and was a really big problem on Oahu. I don't know if any of you were familiar with it when it first got here. But the nice thing is that there are a lot of natural predators and parasites in our state that feed on these 4 different insect groupings that we spoke about. And they tend to, here's an example, they tend to keep these populations really low. with the giant whitefly, when it first got here, it took off, right? Because it was like, wow, this is paradise. And the good news is that it brought its parasite with it. So it came in from Florida and it brought its parasite with it, and that just knocked it back. Once the parasites got established, it took time for them to catch up, right? Because they needed something to feed on. They need their host before they can become their parasite. So, what happens in the other extreme, though, is that we get no parasitism. Here's an example of what the parasitism would look like. All these pukas here tell you that the parasites have emerged. And if they have no parasites, then it just gets covered. The plumerias just get covered. And in this case, the, uh, what are they? I guess the owners of the property on these resorts were complaining that the sugary substance, the honeydew, was dripping down onto the cars. And so what we determined is that they were spraying indiscriminately with pesticides, and that just knocked back the parasites. It did nothing for the pest. And we'll talk about that a little later. So first off, getting back to that sooty mode, this is often the result of one of these insects, the aphids, the mealybugs, the soft scales, or the whiteflies. As they are feeding again, they're releasing or expelling all this sugary substance called honeydew, which drips down on the foliage and causes or allows for the growth of that fungal grouping, that dark fungal grouping that coats the foliage. A good way to test if that's what's going on is just using a piece of Scotch tape, and it'll lift right off. So the plant isn't harmed by the sooty mode. It's harmed, of course, by the insect, which is feeding on the lower leaf surface. So if it's all this sooty mode growth here on the upper surface, that means that the leaves above it on the lower surface have the insects, and it's raining down on these leaves and allowing for the growth of the fungus. But once you control the insect, the sooty mode simply curls up and falls off. Of course, the other problem is that the sooty mode can cause a reduction in photosynthesis, but by far the insect is your bigger problem. So we talked about these biorationals, and I tend to get ahead of myself. I get excited and I forget to look at my notes, so I'm going to try and stick with them. So although management recommendations are best when they are based on a specific pest and its host, considering the label restrictions, of course, an approach incorporating the use of many management strategies, such as biological, cultural, physical, chemical, will prove to be the most effective. So how do we do this? Well, you want to start clean. Uninfested crop plants, right, will allow for the plant to become established in the landscape and better able to defend itself against pests. Remember, prevention is your first line of defense. Healthy, nurtured plants resist pests and diseases better than weak, unhealthy plants. And sometimes the weak, unhealthy plant needs to be identified. Why is it weak and unhealthy? What's growing on? And we kind of talked about that a little bit, about some of the cultural problems, right? Remember, don't love the plant too much. That will set the plant back when you bury it, or you plant it in the wrong area, or perhaps there was some pot-bound issues, and then the plant struggles, and of course it's just a magnet for all these different pests. So you want to keep the area free of weed hosts, which can be serve as alternate hosts for insect and mite pests, right? They can build up on that, as well as diseases such as the viruses, and that will help keep problems to a minimum. Sometimes sacrificing a heavily infested plant in the landscape to spare the less affected plants and replacing them with a healthy plant will keep problems at a minimum and reduce your costs. You won't have to be trying to battle back this pest that may be resistant to what you're spraying. Monitoring the landscape for problems regularly helps identify pests early and increases management options available to you. If you wait until the infestation is too high and then try and battle it back, you'll find yourself in a losing battle. So remember, Hawaii has an abundance of biorationals, okay? Parasites, predators, and diseases of these insects and mites, and these diseases and pathogens. Okay, so there are diseases of other diseases, if that makes sense. When we indiscriminately apply a general insecticide, these parasites and predators are adversely affected, and we see an increase in pest populations, like I explained on that spiraling whitefly. So an effective management program includes pesticides for prevention and active treatment, okay? But beginning with the least toxic pesticide available, okay? So soaps and oils, these biopesticides, right? Botanicals, like Botanigard, Bavaria or something. You want to start with stuff like that before the applications of synthetic general pesticides, because you want to try and preserve these beneficials. And it also reduces the pesticide resistance. Unfortunately, sometimes we have to resort to the pesticides, and then you want to choose the best one. And we'll talk about that a little later. So I thought the next thing we do after we cover those really bad guys is look at some palm pests and disorders, if that's all right. So these are some of the different palm problems I see come through the lab from throughout the state. This is a really common one. We've probably all observed these symptoms on a coconut, and it's caused by a very tiny mite pest. This mite is 1/100th of an inch, and they actually call them worm mites because they really do look more like a worm than they do a mite, right? All the legs are up here at the front of the head, and then they just drag their abdomen as they walk around. They're called Europhiid mites. And the damage symptoms are brown scarring of the and necrosis of the nut, cracking. The nuts may not mature, and you can see here that they simply abort off of the raceme, off of the nut structure. There you go. How's that? Okay, so the Europhiid mite is feeding under the hard petals and sepals. Okay, so they're under here feeding, and they do not come out, and that's because they're very prone to drying out. So we'd never find them out here. They're up under the calyx here, and if we peeled it back, we'd find it. So they're feeding as the nut is developing and growing, and so that damage becomes evident as the nut expands. So it's not very obvious. It takes a high-powered microscope to see them, but the damage is pretty obvious. Okay, coconut leaf roller is also rather common on the palms here. This tends to be a bigger problem along the coast, the windswept areas. And the reason for that is that we believe the reason for that is the parasites have a harder time navigating to find the pest in those windy areas. So the young larvae feed on the underside of leaves beneath a protective thin web of silk. They fasten the lower edges of the leaflets together with ropes of white silk, providing themselves with a protective covering like a tent, while they eventually skeletonize the leaf. We can see that here. So when you peel open these webbed together leaflets, you'll find not only a caterpillar, but you can get lucky and find the pupae, and then this is what the adult moth looks like. So usually well maintained by parasites. You won't need to do much. Of course, if you have a tall tree, pesticide applications would be difficult. If you are able, the bacillus works really well. The bacillus thuriside, thuringiensis, like thuricide, will work well. A banana moth is a common problem throughout the state now. They feed voraciously and are most often attracted to plants growing under stress. Once established, they will feed on healthy tissue and kill the plant. So when insect or a disease is attracted to a plant that's stressed uh, and weakened in some way and then moves in and starts to kill it, we call that an opportunist, or for its ability to kill or feed onto healthy tissue from dead, dying tissue. And this is one of those insects that will do that. So this here is a Pichardia. It was really under stress, great deal of stress. The banana moth moved in, began feeding on the dead tissue here, and then went in and killed the terminal. So some of the different options available, this is one of those insects that the parasites have a real hard time with, and that's because they're feeding down in this terminal. So not much access for these parasites and predators. And so we've got to do something a little different. And some of the strategies include applying the products like pyrethroids, such as bifenthrin, at 2-week applications, and that seemed to manage it well. Unfortunately, the BT, that Bacillus thuringiensis, was not effective because it is a stomach poison and it needs to be ingested, and so they're not able to get that product down into this tight areas of the plant for control. Okay, so we do have some weevils in palms here in Hawaii. There's a whole bunch more throughout the nation and the world that aren't here yet that we are worried about. The ones that are here include this New Guinea sugarcane weevil, and this recent introduction into Hawaii, this silky cane weevil. So this New Guinea sugarcane weevil is found to be a pest of sugarcane as early as 1865 in Punene, Maui. And let's see, does it not come up? There we go. So you can see that they can be quite variable in color. This here coloration has a bunch of different macrolynid mites on it. And these mites are simply hitching a ride on the weevil. They're primarily detritus feeders or rotting vegetation feeders. Some are predaceous, but in this case, they're probably just hitching a ride and moving from plant to plant as the weevil moves around to feed. So they're not actually parasites on the weevil. The silky cane weevil, or the West Indian cane weevil, was first recovered northwest of Hawaii Island in 2016. The adults are about a half an inch in length and range in color from orange to black, and has a slightly variable pattern. They do stand out with that bright orange color. Hosts include sugarcane. Preferred host is the preferred host. Also on palms and bananas. Last month we found it on a Lo'olu palm here in Hilo. So it is starting to spread around. You might want to be aware of it over on Maui. Yeah, unfortunately, management can be very difficult because careful inspections and strict sanitation will actually prove to be the most valuable way to control the problem. And that's because once they get into the palm and do this much damage, there's no recovering for the palm. It's very difficult to get the palm to recover once they've bored into the trunk like this. So Bavaria might be an idea. Things like Botanigard, Bavaria bassiana, might give you control if you're able to contact the insect. Of course, in situations where they've bored or tunneled deep into the palm, that won't be true. So things like pyrethroids, bifenthrin, or maybe a systemic like an imidacloprid might give you a little bit of control. But if the tissue is dead, then there won't be much vascular movement. So that will also be difficult to control. The other thing is that this new silky cane weevil, in some literature, actually implicates it in the spread of a nematode, which causes this red ring disease. It's primarily vectored by the palm weevil, which we don't have in Hawaii yet, but unfortunately that's on the US mainland. And we worry about it getting here. This nematode red ring disease has not been reported in the US, so make that understood, but it is in South America, and it's rather devastating on coconut. Okay, so this is another pest that you've all heard about. It's been in the news quite a bit. First recovered on Oahu in 2013. It's not been recovered on any of the native neighbor islands. The native range includes Southeastern Asia, China, India, Japan, Philippines, Sri Lanka, Thailand, Vietnam. It was introduced throughout the Pacific Islands, primarily as a result of the increased sea traffic during World War II. So it was present on Guam, and that's how we got it here. The larval hosts include rotting coconut, breadfruit, kamani, mango, hala, any other rotting organic matter, including steer manure when it's laid on the ground to compost, so they can get in there and feed on that. That's what the larvae feed on. The adults feed on the young terminals of coconut and African oil palm and other palm species, including areca, bamboo, bottle, date, fan, fishtail, foxtail, pandanus, sago palms. So rather wide host range, but you can't miss them because they're pretty large grubs. You know, an immature beetle is called a grub, not a caterpillar, but the grubs will bore, or the adults will bore into the terminal and feed on the coconut, whereas the grubs again are down in the detritus. So if you're working in your compost and you see these large grubs, then you should be concerned. And yeah, so we want to monitor for that to make sure it doesn't spread around. Here's an example of the types of grubs that you might come across in your compost. We can see things like that New Guinea sugarcane weevil, the flower beetle, a Japanese flower beetle, Asian flower beetle, I think. And then the coconut rhinoceros beetle is a lot larger a grub. So pretty large grub. Oh, here it is. It's the Oriental flower beetle, sorry. So these another way to help you identify it is that these tend to roll over on their back and kind of move around when you lay them out on yourself, on your hand, so you can kind of differentiate them from the rhinoceros beetle. But again, they are very large grubs. Here's an example of some newer pests publication that we put together on coconut and palm pests to help you keep an eye out and help us monitor its their entry into the state. Unfortunately, it is dated. It was published in 2012, and in 2013, I said the rhinoceros beetle became established on Oahu. They're still working on trying to fight this back, but because of that wide grub host range, it's proving difficult. The other weevil that we really want to monitor for is this red palm weevil. This guy has made it to California, so we're worried about its presence or its introduction to the state. And this red palm mite, we're worried about that as well, and keeping our eyes open. This Queensland longhorn beetle, I'm not sure if you've all heard about it there on Maui. Unfortunately, it's the talk of the town here on the Big Island, on the east side of the Big Island, where we first began seeing it attracted to people's screens, window screens at night, because it got attracted to light, and we could never find a host. We believed it was feeding on some of the rubbish, or I should say quote unquote rubbish trees in the forest like Albizia. But it eventually has become established, and it has been found feeding on kukui, breadfruit, various citrus, sagos, cacao, trumpet tree, calamungai, that moringa oleifera. It's native to Australia, where it's apparently well managed biologically. Um, the Department of Ag, who is actually charged with looking for biocontrol agents for the state, is doing some exploratory work to see what they can find there. The management is very difficult, as you can imagine. Once it bores into the trunk of a tree, um, uh, it's hard to control, and a hole this size actually will cause quite a bit of damage to the trunk and allow for secondary invaders. Remember that term I said, opportunists? So yeah, these opportunist organisms will move in and start to decay the tree. And so control is rather difficult. So prevention, sanitation, monitoring the plant is what we're suggesting. And even prophylactic sprays of an insecticide to try and protect the tree is a good idea. Okay, let's take a look at some diseases. This ceratocystis or Phalabiopsis paradoxa is a rather common pathogen in coconut culture here. We can find it on different palms, but it tends to be a bigger problem on coconut. And that's because a lot of our palms in landscape, or coconut palms in landscaping, are transplanted. And they're large, they're difficult to work with, and so they become injured. And it's these injuries that allow for the entry of the organism. So what are the symptoms? Well, it's usually a soft, slowly expanding decay of the trunk tissues, darkening to black with age. As the decay progresses, dark, a darkly pigmented liquid bleeds down from the trunk, from the point of invasion. Okay, so it starts to what we term bleed out of the wound. And that's the plant sap. The plant sap exuding from the tree. Often trees and different plants will exude sap to try and repel or push out the infection or the insect. And so the plant starts bleeding like this. You can see that you've got an active infection going on. We'll see all the damage right on the trunk of a tree, but they don't bleed, and that's because they're not infected. But once they start bleeding, you know that they've got an infection in there. So in advanced cases, the interior of the affected trunks are hollow due to the decay of the interior tissues. The palms are monocots, so it's just all full with vascular bundles, and those vascular bundles are not directly connected like in a dicot, so that infection is a little slower to progress around the trunk, but eventually it will, and it will actually destroy the palm. So roots may be similarly blackened and decayed. The disease tends to be a bigger problem up in the less lignified area of the palm. So it's not as hardened off yet. The green woody tissue, the green tissue is less lignified. So when we come in and we actually prune the palm, we can not only cause that wound, but we can introduce disease. And so often we'll see the disease progress from here. But as I said, they're often transplanted and wounded in the process, or as they are harvested from the ground, the roots are pruned and wounded, and the organism moves in that regard, in that way. The ceratocystis paradoxa is a common sugarcane pathogen, so it's pretty ubiquitous in Hawaii's environment, and therefore the organism simply moves around and is present um, in quite a few areas And so moving the palm that's been growing in this type of soil will probably introduce the organism. So what do we do? Well, avoid wounding the palm trunks at all stages of growth and during transport. Especially here on this island, and I think it's true on the other islands, is a lot of our coconuts that are in the landscaping at the hotels on the west side of the island are from here on the east side of the island. And we of course grew a lot of sugarcane here on the east side historically, and so the soils have the organism. You also want to avoid wounding the stems during landscape operations, weed trimming, tree trimming, weed whacking. We suggest sanitizing the tools between trees as you remove these leaves so that you're not spreading the disease from tree to tree. You want to cut and remove and destroy the severely diseased trees, as they may serve as a source of disease for neighboring plants. In some cases, early or localized infections can be excised through tree surgery, followed by thiophanate methyl treatment, which is like what was the active ingredient Benomil. That's now available in products like Cleary's 3336 in other formulations or other approved products. But you need to be careful, of course, with that, because in a landscape situation, if the damage is too severe, they become fall risks. And, of course, you wouldn't want the tree falling on a structure or a person. Early bud infection or leaf and stem tissues can be effectively treated by this thiophanate methyl, and avoid direct impaction of sprinkler irrigation upon the wounded stems, right? Because you've got the benefit on the west side of the islands, on the leeward sides, to have less rainfall than we do here on the windward side. And so if you are able to manage your irrigation well and not wet the palm trunk, you help to reduce the infection. Fungi need moisture in order to cause disease. And then as you receive new palms, you want to be sure that they're nice and healthy and clean. Okay, so
Brian?
yes.
Brian?
Yes.
Hey, thank you so much. You're doing a great job. I just want to interject. It is 10:11, so you have about 20 minutes left. And then we have questions and answers. I know some people have to leave around 10:30. If not, there's no rush. You can always stay later, but I just wanted to give you a time check.
Thank you, Hannah. Yeah, I tend to talk too much. That's what my wife says, at least.
No, it's really very informative. Thank you so much, Brian.
So, yeah, I see that I've got, what, am I halfway even? Not quite. Just about halfway. So, how would you like me to handle this? Do you want me to just breeze through it, or just continue and people leave as they will, or what's the group?
Maybe just try to speed it up a little bit, and then people can ask questions in the chat, and we can still have a question and answer session at the end.
Okay, I'll do that. Yeah, I tend to start thinking about all these problems or samples that have come through, and I've got a story to tell, so I'll try to cut that out. Okay, if we look at some other diseases of palms, they include phytophthora diseases, and they can cause a bunch of different symptoms ranging from root rot, bud rot. The terminal spear on coconut trees is rather common, and it can be characterized by these symptoms on the nuts. The nuts simply drop off the tree. Pestalotia palmarum is also a problem on palms. They tend to cause this type of necrosis of the foliage. So this looks really different from that coconut leaf roller, right? The leaflet is still intact. Nutrient deficiencies are also a big issue with palms. They can be quite characteristic, especially when you look at boron, which causes this type of crinkled and unusual growth. Potassium, magnesium, not quite as easily identified, but you need to be careful about how you're fertilizing your palms. You want to use something that's got a nice general balanced analysis with the minors. So let's talk about turf pests and diseases. Turf caterpillars, we really have three moth problems here in Hawaii, and we've also got one butterfly, the fiery skipper. They tend to be a problem, at least here on this island, during the late summer, early fall. They're very common. The lawn armyworm, the grass webworm tends to be the most destructive pests here on Hawaii, and then a bunch of different cutworms as well. Sometimes you can do localized spray applications to knock them back. Otherwise, you've got to go with something broad spectrum, and perhaps a granular insecticide will help to manage them. Foliar turf diseases, there's a bunch of different ones. They can be stress induced. I wonder what happened here. Looks like it was caused by the mower, as they drove across. But they can actually cause quite a range of symptoms that are rather diffuse and not so easily identified, requiring sample submission to identify. But usually general fungicides work pretty well at management. Stolen turf diseases tend to be a little more easily identified. Things like fairy rings, of course, really easy to identify. Large brown patch, these rhizoctonia diseases, yellow patch and other rhizoctonia, also easy to identify. Take all patch, it kind of gives you this, they call it a frog eye, where you've got these weedy things growing in the center because it's killed the grass. They produce this type of structure, which is visible under a hand lens, not so much this hypophodia, but certainly the darkened hyphae, southern blight, such as sclerotium, is also rather easy to identify, as is the pythium, because of the mycelium or the fungal strands. Nematodes can be a big issue in cropping systems, not even a millimeter long, but still able to destroy your landscaping. These microscopic worms infect the roots of plants and suck nutrients and water out of them. Nematodes feed on a large number of plants, if not all plants, and cause billions of dollars worth of damage to world food crops, as well as tree crops, turf, and ornamentals. Nematodes must always be considered as a possible cause for a declining or damaged plant. There can be either an ectoparasite, which means the entire body of the nematode is inside the root, or an ectoparasite, where they feed from outside the root and only their heads and their mouth part, called a stylet, is in the root feeding. So they can cause a bunch of different symptoms on the roots, causing a foliage symptoms, but foliar symptoms, root feeding causing foliar symptoms, but they can also be a foliar nematode, which means that they do not live in the roots. They simply live up in the plant, up in the leaves of the foliage of the plant. So like on ferns, peperomias, or even on orchids. Turf parasitic nematodes are pretty obvious damage, especially if you're looking at, the damage symptoms in the in a golf course, not so easily identified, really diffuse, kind of looks like a number of other problems. But of course, once you lift up the roots, you'll be able to see if you have a problem. In the case of a root knot nematode, the roots get kind of gnarled up and knotted. And in a case of another nematode, such as a lesion nematode or a spiral nematode, or even this ring nematode, the root system is much less developed or not as healthy. This is a left side doing nice, right side not so good. I hope I'm not forgetting anything as I'm rushing here, but we do have a publication on these plant parasitic nematodes and their management and how to properly sample for them. This here is a trial of a Bayer product. Bayer actually created a product developed a product called Indemnify, which is available for turf. And it's the active ingredient is fluropyram. Here we see how well it worked in untreated and a treated right down the middle. So this product is actually now available for ornamental crops as well. I can't remember offhand what it's called, but we did some trials on anthurium and it worked really well. So turf mealybug, scale insects, also a significant problem here in the state. This Rhodes grass mealybug is probably one of the more common ones. Rice whitefly is also an issue. Oh, mealybug scale, I didn't write the whitefly. Okay, so this whitefly is also a big issue here. This scale, no common name, is recently introduced. We found it here on the west side of the island, and it has since spread around the whole island, so it may be headed your way. This is on Tropic Lalo, a ground cover, and it simply turned it snow white. It was so heavy. Bermuda grass scale is a problem. It's down into the scale under the sheath of the leaf on the internodes. And the Tuttle mealybug is also present on the foliage. Miscellaneous landscape pests. The monkeypod caterpillars you're probably familiar with. There's 3 of them that are a problem. They can be managed quite well with pesticide applications made to a structure wrapped around the trunk of the tree. Cotton lacebug is also a problem on a bunch of different crops. Let me see if I can included soursop, orchid tree, coral tree, the willy willies, hibiscus, mulberries, just a whole huge range of host families, Fabaceae, Malvaceae, Annonaceae, Solanaceae. The symptoms are pretty obvious. Their mouth part is like a stylet, so they actually are sucking out the juice of the host as they feed, giving it this white stippled look. Azalea lacebug is also another lacebug in Hawaii that's a problem, but tends to be a little more specific to its host range, which are evergreen azaleas, rhododendron, parantha, Andromeda vireas are the preferred hosts, usually other members of the laurel family. This cypress or Pacific beetle roach is phytophagous. It's the only known species that are viviparous, which means that they have live births. The young are nourished inside her body until they're big enough to defend themselves. So have you heard of cockroach milk? Well, this is where it came from. It's the latest superfood. Maybe it's not, it's more than 4 times as nutritious as cow milk, but it's missing amino acids we need for human development. So while studying these roaches, scientists discovered milk crystals that they feed their developing young. So it's pretty cool in a kind of a nerdy entomologist way, but they are phytophagous, which means that they are feeding on the trees and they can cause this type of symptom. They tend to be down under the tree, living in the leaf litter. And so that's the way to manage them, is targeting that area. Little fire ant is a issue that we all know about now because it's spread throughout the state, and it can be a big issue here on the island where folks don't even want to go into their crop and work unless it's managed. This nettle caterpillar, when this first entered Hawaii, I found it on palms that were introduced to the state properly through the USDA, but as you can see, the eggs are hyaline. You can see right through them, so they weren't noticed. And unfortunately, once the grower made contact, it had spread into the forest. And the grower grew on 2 sides of the island, so it spread throughout the island before we could actually jump on it and do something. The good news is that, well, it was really bad, right? You see here how severe it was. Even the kids wouldn't play in the lawn because they're all in the grass. But the the Department of Ag actually went to Taiwan, where it came from, and found the parasite. And that parasite took such good care of it that it keeps it at very low levels. I did want to point out this slide though. When we look at biocontrol, we need a pest. So as the pest population will build, so does the parasite. And then it's killed off all the pests and the parasites crash along with the pest. And then of course the pests take off again, right? And the parasite has a lag because they need the pest to feed on. And so we'll see this type of activity with this nettle caterpillar here, but at much lower levels than it was prior to the introduction of that parasite. This bagrata bug was on Maui first, and then it moved over here in 2015. It feeds on cruciferous crops. This here is, let's see, it's Mayapilo, which is a crucifer native plant. This is at our Honokohau, Kaloko Honokohau National Park on the west side, and it actually killed the plants. So it can build to really high levels. Looking at some viruses that you might come across, this T ring spot virus was first observed on Oahu in 2009, and then here on our island in 2012. Dr. Melzer identified it to be an Emarovirus, which is a T ring spot associated virus. So that it's an associated virus, and it's discovered, what we've recently discovered and what we found is that we also have a couple of new Eryphiid mites. Remember we talked about the Eryphiid mites earlier, these worm-like mites where the legs are on the front and they drag their abdomen. Very tiny mites feeding on the tea leaves. They also are brand new to Hawaii and we don't have an ID on them yet. But this ring spot virus is being found in the mite, so we believe they are the vector. Okay, so a new viral infections of gingers has also been recently identified. The symptoms can be found on flowering ginger plants infected with a single or multiple combination of the viruses. However, only plants infected with the podivirus, which is the banana brack virus, show clear and easily identifiable symptoms. Symptoms of disease in red ginger are difficult to determine when infected with the two badenoviruses, the Canna yellow mottle virus and the banana streak virus, which I don't have a picture of here. Anyway, yeah, mixed infections are rather diffuse, but the banana brack will show up as an observable infection. So the banana brack is spread by aphids like the banana and melon aphid. Canna is Canna yellow mosaic by mealybugs such as citrus, pineapple, or banana mealybug. Banana streak symptoms are not clearly defined and we're not quite sure, but we believe it's spread by mealybugs. And then we're also observing a dieback, which is also not clearly defined just yet. Heliconias and floral gingers can also host the banana bunchy top virus, which is spread by aphids. Okay, so we were going to talk about pesticides. And although management recommendations, again, are best based on a specific pest and its host plant, an approach incorporating the use of many management strategies will prove to be the most effective, right? So, we're looking at things like regulatory control, which would be in inspections and embargoes, cultural control, crop rotations, host plant resistance, cultivar improvement, right? Traditional breeding or genetic modification, biological control, use of these parasites, mechanical control, physical control, biorational biopesticides, even things like insect growth regulators, and of course, chemical controls. And these different chemical controls on different crops is available at these two websites. So disease management, let me see, I know we don't have any time, Hannah, but I do want to make sure I stress a couple of things if you will let me. and that is the best disease management strategy employs a variety of physical, chemical, and biological methods, just like with insects. So it's an integrated approach. Physical methods include sanitation, using these physical barriers and other steps to exclude pests from growing environments, working the diseased or infested areas last, right? So growing in greenhouses, on raised benches, pathogen-free plants in pots, potting media can greatly decrease the spread of a plant pathogen. The key to physical methods for disease control is to keep the crop from becoming infected. Once that happens, other less effective management strategies, such as fungicidal applications, must be relied upon. So if you do rely on those things, you won't be very happy with your results. And biological methods includes the use of pathogen-free plants. When you use propagative material that is infested with a plant pathogen, virus, or other infested with arthropod pests, you only ensure that you'll have the pest problems throughout the life of the crop. So no matter how good a job you do at preparing the landscape or nursery with clean pots or potting media, if you use infected plants, you've negated all the benefit of your hard work. And of course, chemical methods are commonly relied heavily upon, right? With like, well, what do I spray? Well, if you want to bring a quality to a quality plant to market or maintain the landscape. However, solely relying on chemical control measures usually gives disappointing results. The first and most important step to pesticide use is a diagnosis, because you want to use the proper product that's going to give you the best control. Um, uh, and that can only be done through proper identification. So looking at some recently identified pests, we'll just skim through it. You're probably familiar with them. This avocado lace bug is really new. We just found it late last year here on the island. I believe it's now throughout the state. I'm not sure if it's on Kauai yet. This Biltothrips causes this really unusual pattern. This is an avocado leaf, the lower surface, as the feeding trails. They get on taro. The thrips you can see the thrips there. And the privet mites on Oahu, it causes this, again, this type of irinose, which is because it's an irophyid mite. So some invasive plant diseases. This is tomato yellow leaf curl virus, this taro vein chlorosis virus. We're not sure what's vectoring it. The rapidohea death, of course, we've all heard about that. Powdery mildew of mulberry is common, and we just found this powdery mildew of lychee. So I think maybe we'll just skip all this, huh, Hannah? Or add it to the questions, perhaps. So if you'd like to guess what might be causing this type damage on hibiscus, this is a gimme, I think. You guys should know this. It's a hibiscus aurelia mite. This here is a crinum lily. I'll get this quite commonly, and people think it's a virus or perhaps a rust disease. But if you look really closely, there's some critters there. Up close, you can tell, well, they're thrips. So this is actually the banded greenhouse thrip that causes this damage. How about on this tea leaf? Can you tell what might be going on there? Or this plumeria? Looks pretty similar. Well, this is Cercospora leaf spot on the plumeria and plumeria rust. One way to tell would be to just simply wipe your finger over it. And if it transfers the yellow to your finger, it's probably a rust disease, whereas the Cercospora won't. Now, things that you should have available to you, things like handheld pH and EC meters, especially on the leeward side, we tend to have more of a salt buildup in our landscaping. Hand lenses, those are cheap and easy to find, and that gives you a better way to measure and take a look at what might be going on. Measuring scales, if you're measuring out your pesticide, and then references. There's a number of really good references available, not only for problems in Hawaii, but also incoming or potential problems. And then our CTER website has quite a number of really great free resources that you should take advantage of. So if you did want to start looking at some websites, I challenge you to look for .edu websites or websites that .gov that have resources and has been actually published and gone through a strict um, a check. So our Hawaii Pest websites include things like our crop knowledge master. Department of Agriculture puts out some great ones. And then, of course, you always want to check your pesticide labels and MSDS sheets. And if you do go to any of the other college websites, I suggest sticking with either Florida, California, or Texas along the in environments that are similar to ours. And with that, I say mahalo. This is our newest beach on the Puna Coast from 2018. So, Hannah, we're going to take some questions, I think, right?
Yes,
I'm out of
thank you
breath.
so much, Brian. That was such a great presentation.
I'm out of breath.
I'm going to look. You did a great job. I'm going to just look through the chat for questions.
One question I could ask is, you know, if avocados
Brian,
Yes.
do you know if avocado lace bugs are on Maui?
I believe they are. I'm not sure if they're established in the environment yet. We haven't seen that, but we know that they were located in the big box stores as plants were shipped from our island.
Yeah.
That's how they've been spreading around our island as folks buy them and plant them in their yard.
Yeah.
Good to know.
What's a good control method for tea ring spot?
What's a good control method for tea ring spot virus?
Yeah, so viruses, once they become established, they're pretty difficult to eradicate. They are still working the details out of that virus. Right now, they look like it could be a localized virus, which means that the virus doesn't really migrate throughout the plant, but they're not sure. And so we haven't really looked at if pruning and removing the symptomatic leaves will get rid of it or not. So it's still in process.
Okay, thank you.
Yeah.
What's a good treatment for?
Just got a comment. Awesome. We love a part 2 video. Mahalo.
One other question, Brian. What pesticide is good for getting rid of soil mealybugs? And how can you tell the difference between mealies and soft white scales?
Between mealies and, I'm sorry, soft scales? Is that what you said? Okay. Well, mealy will move. They actually have legs that allow them to move around. So if you were to poke them with a tip of a pencil, they'll kind of boogie around, whereas the scales won't. So that's a way to differentiate. As far as pesticides, you know, Dursben was great for controlling these root mealybugs, but of course it's not available to everyone anymore because it's a restricted use product. And so one product that seems to work pretty well are the pyrethroids like bifenthrin.
Okay, thank you.
For if I could expand on that, for management of foliar mealybugs and scales, the IGRs worked well, which are the insect growth regulators like buprofezin or pyroproxyfen, and that could give you some control of the root mealies as well.
What was the question?
What's a good treatment for pink hibiscus mealybugs?
Brian, what's a good treatment for pink hibiscus mealybugs?
Okay, so I guess I can review or repeat that. The IGRs work pretty well against these soft-bodied insects like the mealies, the scales, and the whiteflies. And these IGRs are insect growth regulators. And products that come to mind are pyroproxyfen, like Distance, or buprofezin, like Talus.
So,
the thing to remember is that that white covering is a wax. Hello?
What'd you say? Sorry.
Oh, That white covering is a wax, and so you want to make sure that you're adding in a surfactant or a spreader so that the pesticide will come in contact with the insect, unless the label tells you not to use a surfactant. But especially if you're using a contact pesticide like bifenthrin, you'd want to be able to make contact with that insect.
Allison, can you repeat what?
Somebody had previously asked if coconut rhinoceros beetle was on Maui.
No, it's not.
Yeah, coconut rhinoceros beetle is not on Maui.
But we want to make sure that everyone is aware so that they can help us monitor.
Right. I also see another question. Can you test soil samples for nematodes?
Yes. And
Yes, and
again,
you can.
please go back to that reference and look at proper way to collect a sample, because that would give you the best result.
Juvenile whitefly look like scale?
Does juvenile whitefly look like scale insect?
Yeah. Yeah, when they're in the pupal stage, they can be to an untrained eye, they could be mistaken for scale. But they usually are in the presence of the adult.
Great, thank you. Is there any other questions?
Here's one. How do you identify two-spot leafhopper?
Brian, how do you identify two-spot leafhopper?
By the adult, I guess, for if you don't have access to the keys and the scope, it would be by the adult. The adult has these 2 spots on the hind portion of their wing. There's some nice pictures online.
Can you test compost samples for pathogens?
Hey, Brian.
Yeah.
Can your lab or the ADSC in Oahu test compost samples for pathogens?
Okay, so that when you start looking at soil and pathogens, it becomes difficult because there's just so much saprobes, right? All these saprophytes are in there breaking down the compost, doing what they're supposed to do. And so if we were to take that compost and plate it, put it into agar, a whole bunch of things would grow. And so the way we work around that is we use augers that are very specific to allow only certain things to grow. So if you're worried about fusarium wilt, perhaps you're going to grow koa, then we'd use an auger specific for fusarium. But to look at all pathogens, it's not possible. It would just, imagine what's growing in there, right? So the ones that we commonly look for are the water modes like Pythium or Phytophthora, and we certainly can do that.
Thank you, Brian, very much. I really want to express my gratitude. I appreciate your time and your expertise and knowledge. I learned a lot from you today. I
Good.
appreciate it.